Mine should be on as well.
Okay, well, we're up for exercise number 2. This is Andrea Wheeler, and she's going to be speaking on digital PCR enables sensitive detection of latent AIDS viruses. She earned her undergraduate degree in biology at Colgate University and has an MS in cellular and molecular biology at the University of Madison.
Yeah,
University of
here.
Wisconsin-Madison.
Yep.
Go Bucky. She has been working at University of Wisconsin since 2006 and in the Virology Services Unit at the Wisconsin National Primate Research Center since about 2010. Andrea, take it away.
All right. Well, thank you, Lenny. Thanks for the introduction and for inviting me here to talk a little bit more about digital PCR. So actually, I think this works really great because Holly talked about all the concepts I think I will mention in my talk, but hopefully you will see that I've been able to really take advantage of some of the features of the absolute Q here. And I swear she didn't put me up to this, but I have some nice data that really emphasizes what she showed you earlier. So I just want to begin by way of introduction of my group. So as Lenny already said, I'm part of the Virology Services Unit at the Primate Center here, where our mission is to provide expert support to investigators conducting virological studies in non-human primates. So a lot of the work we do is actually measuring viral burden in samples collected from animals that are part of studies or from in vitro experiments. And we mostly do this using qPCR and RT-qPCR. Over the years, we've developed a portfolio of viruses that we measure, which now includes both RNA and DNA viruses. We provide this quantitative viral RNA and DNA testing from a range of samples, including plasma and serum, blood, urine, swabs, tissue culture supernatant, a variety of fluids, as well as from biopsy and tissue samples. Traditionally, all of this work has used real-time platforms, so lately we've been exploring digital PCR for certain assays, particularly those that require extra sensitivity for detecting rare targets. Beyond testing for viruses, the Virology Services Unit also produces characterized virus stocks that can be used for both in vivo and in vitro studies. Most recently, we've produced stocks of influenza, Mpox, Clade 2B, and SIV. Coincidentally, it's SIV that I'm going to talk about today. Virology Services actually has a long history with SIV. We got our start monitoring SIV burden in animals that were part of the many SIV studies that were taking place at the Primate Center. It's also a point of personal interest for me because I did my graduate work in the HIV and SIV field. So as therapies and treatments for HIV have improved over the years, there's been increasing demand for exquisitely sensitive assays for detecting these AIDS viruses, and in particular for quantifying the latent reservoir of HIV and SIV, which is the true barrier to cure. That's what I'm going to focus on today. I'm going to talk a little bit about reservoir assays, the technical challenges, and how we use digital PCR to quantify the latent reservoir. So because it's going to be a little while before I actually get to talking about digital PCR, I just want to give you a brief idea of where this talk's going. I think this will underscore how using digital PCR on the absolute Q system provides a platform for measuring a rare species of cells that are latently infected with SIV, and doing so in a sensitive and easy to perform manner. So to really appreciate why this is remarkable, there's some things that you will need to know. I'm going to begin by briefly giving some background about SIV, then talk about why we need reservoir assays, and what the technical challenges are to performing them. I'll mention some existing reservoir assays, including the Integrated Proviral DNA Assay, or IPDA. Then I'll explain how droplet digital PCR and digital PCR are uniquely suited to perform this assay, and how using the absolute Q rather than performing droplet digital PCR has allowed us to make some improvements to this existing assay so that we can perform it more easily and in a more time and cost-efficient manner. I assume most people here are familiar with HIV and maybe the monkey model for studying HIV, simian immunodeficiency virus, or SIV. You probably also know these viruses are retroviruses, which means they're RNA viruses that go through a DNA intermediate to replicate. We call this viral DNA provirus. Interestingly, these viruses actually integrate into the host genome and take advantage of the host machinery to produce more virus. This means that virus can actually persist in two ways. First, once integrated, it can replicate and produce progeny virions. During active infection, this is the major source of virus. But also, since the virus integrates into the cellular DNA, in the absence of active replication, the viral DNA remains stably in the cell undetected. When that cell divides, the viral DNA is then passed along to the daughter cells. HIV and SIV have genomes that are just under 10 kb long and encode 9 proteins. These genomes also have long terminal repeats, or LTRs, on either end of the genome, here and over here. I do want to mention that now because it is going to come up again later in the talk. And then finally, the primary cellular target of the virus is CD4 positive T cells. So to illustrate why we need reservoir assays and the challenges to performing them, I first want to talk about the normal course of HIV and SIV infection. Early in the HIV pandemic, Mellors et al. Showed that the best indicator of progression to AIDS is the plasma viral load. So this is what we typically monitor. Here I'm showing representative graph of what plasma viral loads might look like over the course of infection. Time is shown on the x-axis and plasma viral load on the y-axis. So following initial infection, the virus quickly enters the body and replicates to a high level. We generally see this peak of viral replication about 1 to 2 weeks post-infection. After that, viral replication is typically brought under control to some level and reaches what we call the set point. This is where viral replication remains steady for an extended period of time before eventual progression to AIDS. There are, of course, many factors that influence to what extent viral replication is brought under control. Some people and animals are unlucky and in the absence of interventions will never really control viral replication and will rapidly progress to disease. This is the line labeled rapid progression on the graph. The majority of people and animals bring viral replication under control to some extent, but virus remains detectable at a steady level in the blood. This is normal progression. There are also some animals and people who naturally control viral replication to levels that are undetectable by our standard assays. We call these individuals elite controllers. But remember, even though the virus remains undetectable, it is not gone. Additionally, HIV therapies have improved significantly over the years. And so through art and other interventions, we can actually achieve these undetectable plasma viral loads. As these interventions continue to improve, there's now the hope of achieving what we call a functional cure, where people could stop treatment without the risk of virus reactivating. But in order to know if these therapies are successful, we need a way to measure the reservoir that remains hidden from view. So before I discuss reservoir assays, I want to quickly talk about what the latent reservoir is and how it is established. As I mentioned, CD4 T cells are the primary target for HIV and SIV. So you can see here in this diagram that activated CD4 T cells are being infected by the virus. Once the virus infects these cells, it will reverse transcribe and integrate into the host genome. Many of these infected cells will die or be targeted for destruction by the immune system as it protects the body from infection. Some of these cells, however, will return to a resting memory state. And they will remain, and there the virus will remain inside the cells undetected. As these latently infected cells divide, the integrated virus is then passed on to the daughter cells. These cells remain in the body with the potential for the virus to reactivate and start producing more virus at any time, which is exactly the problem. The reservoir is always there with the potential to mobilize. I also want to note that it's been shown that latently infected cells are long-lived in the body. So we're now seeing therapies that are designed to target and destroy this latent reservoir. However, in order to know whether they're working or effective, we need a way to measure the latent reservoir, which it turns out is very challenging. Latently infected cells are found at a very low frequency in the body. Estimates of size of the reservoir in individuals on ART, or those who naturally control virus, range from 0.01 to 100 latently infected cells per million CD4 T cells. This makes detecting latently infected cells comparable to searching for the proverbial needle in a haystack, as I know we mentioned a couple times already, but it's a common theme here, I guess. So another thing to know about the latent reservoir is that much of the integrated provirus in cells is actually defective, meaning that through the process of infection and integration, or due to a post-integration event, deletions or mutations have been introduced, rendering the virus incapable of reactivating. So in a study by Bender et al., where they used near full-length deep sequencing of SIV provirus, they looked closely at the amount of intact provirus. In this figure, oh, shoot, now it's working. Sorry, okay. In this figure from their paper, they show provirus by type. So the orange parts of these bars represent intact provirus. Provirus containing deletions of various sizes are shown in shades of blue, and provirus containing mutations are in green. Bender et al. Found that on average, about 28% of the SIV provirus remained intact. And an interesting note is they also showed that in HIV, this number is actually lower. The rest of the provirus contains deletions and mutations of one kind or another. You can see here too that in greens and looking at the greens and blues in this graph, the majority of defective provirus is due to deletions and hypermutation. So the challenge with reservoir assays is that not only detecting that rare proviral DNA, but also differentiating the intact proviral DNA from the replication deficient proviral DNA. This is where reservoir assays come into play. A number of assays actually exist, but none of them are perfect. Some of them overestimate the size of the reservoir while others underestimate it. Different assays also give you different information about the reservoir and how likely it is to reactivate. So I like to think of these assays as existing on a spectrum, and we just need to find the right balance between the estimation of reservoir size, the information we can get, the cost of the assay, and how labor intensive it is to perform. So on one end of the spectrum, here in blue and black, we have PCR-based assays. These assays are generally more cost-effective and easier to perform. They also tend to drastically overestimate the size of the reservoir because they do not readily discriminate between intact and defective provirus. Rather, they simply detect that viral nucleic acid is present. On the opposite end of the spectrum here in red, we have the cell culture-based assays. For example, the QVOA, or quantitative viral outgrowth assay. This assay has actually been considered the gold standard of reservoir assays because it only measures virus that has demonstrated the ability to reactivate. However, it also underestimates the size of the reservoir because not all provirus that is present will reactivate in a given experiment, even if it may have the potential to reactivate. This assay is also quite labor intensive to perform and requires a lot of cells. So the IPDA that I'm going to talk about today exists somewhere in the middle of the spectrum. We think it is a really nice balance of being easy to perform, cost effective, and gives a reasonable amount of information about the proviral DNA that is present. It is a PCR-based assay, but it also includes methods for evaluating whether that provirus is defective or may be capable of reactivating. A quick note is that as I move on to talking about the IPDA itself, I'm going to talk specifically about SIV because I'm employed by the Primate Center where we focus on non-human primates, but the same concepts apply to the assay performed for HIV. The IPDA was actually developed by Bob Silicano's group at Johns Hopkins with the goal of quantifying HIV or SIV that likely retains the ability to reactivate. That means that it specifically enumerates provirus that is integrated and devoid of large internal deletions and hypermutation. The IPDA was actually developed using droplet digital PCR, but I will show you there's some advantages to performing this assay using digital PCR specifically on the absolute few. So I'll discuss details of the assay in the coming slides, but I just briefly want to mention that it enumerates this intact provirus through a series of probe-based droplet digital PCR reactions. One reaction targets two specific regions of the SIV genome, a second reaction targets unintegrated provirus, and a third reaction targets two regions of a host gene. So I'm going to talk about each of these reactions individually. The first reaction for SIV targets includes two amplicons in the SIV genome, one in the OMV gene and one in the PAL gene. The OMV and PAL amplicons were carefully chosen and described in that study by Bender et al. Based on deep sequencing of latently infected cells. This figure from their paper shows their sliding scale analysis of their sequence data that they use to determine which specific regions of the genome are most often deleted. If we look carefully at this graph, we can see, I can't really see where EMV and PAL are, but they show that these specific regions of EMV and PAL are most frequently deleted if there are deletions present. So therefore, if we can detect both of those regions in a particular provirus, it is likely lacking major internal deletions. So this is actually one of the critical features of droplet digital PCR or digital PCR that Holly already mentioned, is that because we're interrogating a single molecule of DNA in each partition, if a given partition is positive for both targets, we know it's present on that same molecule, which is very important when we're looking at intact provirus. This would not be possible with regular PCR. So in addition to accounting for deletions in the SIV genome, this first reaction also accounts for ApoBac3G and F-mediated hypermutation. So for those not familiar, the ApoBac family of proteins are cytosine deaminases that play a role in response to infection. They've been shown to cause G to A hypermutation in both HIV and SIV infections. This is illustrated here where we see that in the presence of ApoBec3G, the Gs have been changed to As. This G to A hypermutation is a significant source of defective provirus, so we need to consider this when we enumerate intact provirus. So this is done by including an additional unlabeled competitor probe in the reaction. The normal labeled probe has the wild type sequence, and that is shown right here. And the unlabeled competitor probe has included the G to A hypermutations. So that provirus with wild type sequence will bind the regular probe and will emit a signal and be counted in our assay. Provirus that contains the hypermutation will actually bind better to the unlabeled competitor probe and will therefore not be counted, will not emit a signal and will not be counted in the assay. So the second droplet digital PCR reaction targets unintegrated provirus. So we're of course mainly interested in provirus that's integrated into the host genome and is capable of replication, but there are in fact other forms of viral DNA that can persist inside the cells. So you remember that I mentioned that the SIV genome has long terminal repeats on either end. These repeats can actually facilitate the formation of 1 and 2 LTR circles. The single LTR circles have been shown to be short-lived and are therefore not a significant source of viral DNA in cells. 2 LTR circles are, however, a different story. They're stable and can exist in the cell similar to how a plasmid does. They are not, however, a significant template for viral replication. So for this reason, we want to exclude this DNA from our analysis. This is accomplished by using a PCR amplicon that targets the 2 LTR junction as shown here on this diagram. This reaction also includes that same OMV target that was included in the first reaction. So we can count the number of partitions that are positive for the 2 LTR junction and those are our 2 LTR circles. So we count those 2 LTR circles, and then when we perform our analysis, we subtract 2 LTR circles from our total intact provirus. So the third droplet digital PCR reaction targets the host gene RPP30. We actually use this assay for 2 purposes. First, we use it so we can relate number of provirus to cell number so that we can report results in terms of intact provirus per million cells. Second, we use this assay to account for shearing of DNA that happens during the process of performing these experiments. As you probably know, DNA can be sheared both during the isolation and during the PCR itself, which means that we could inadvertently skew lower the number of DNA molecules in a sample that contain both the Pol and amplicons. So to account for this shearing, we calculate the DSI, or DNA shearing index. This is calculated using these 2 amplicons in our RPP30 gene, which are strategically placed the same distance apart as the SIV OMV and Pol amplicons, which is about 5 kb. Because we know that in a cell, the 2 amplicons on the RPP30 gene are always found on the same molecule of DNA, we can calculate the frequency at which we see these amplicons detected individually and use this to adjust the SIV targets accordingly. So now we're getting back to digital PCR. So hopefully by this point, I've clearly outlined why we need reservoir assays and how our assay of choice, the IPDA, works. I want to take a moment to reiterate that the latent reservoir is a rare species that exists in a sea of uninfected cells, therefore requiring extremely sensitive assays to detect it. The low frequency of latently infected cells make digital PCR an attractive and appropriate platform. In addition, because of the nature of the assay, regular PCR or even qPCR are not sufficient for the IPDA because they interrogate bulk DNA, which means it would not be possible to know if our 2 SIV targets are present on the same molecule of DNA rather than just in that big pool of DNA. So finally, I want to go on to show you that digital PCR offers several advantages over droplet digital PCR, the platform on which the IPDA was first developed. First, it is much easier and quicker to use the absolute Q than it is to generate droplets for droplet digital PCR, which are ultimately fragile and difficult to work with. I will also show you that we get more data using the absolute Q and that it's much more consistent. So based on my previous experience with droplet digital PCR, I already knew how tedious and labor-intensive that process is. Working with oil and water emulsion droplets can be very tricky. So when I heard that the BioGarage had the absolute Q available for us to work with, I was very excited to try it out. So the first thing I did was just to test out the assay on the absolute Q and make sure it would perform as expected and the results would be clean and easy to interpret, just like when we use droplet digital PCR. So I'm just showing some representative raw data that I generated using the absolute Q instrument. Importantly, so for the sake of time, I'm not going to show you that same data from the droplet digital PCR, but I will show you that the results look pretty much the same. Importantly, regardless of the platform we use, we see clear separation between the positive and negative partitions with very little range, and we get the same proportion of positives from each platform. So here I'm just showing in the top row the Paul, and 2 LTR circle reactions using FAM, VIC, and Sci5 dyes respectively. I'm going to come back to this again in a little bit, but in this experiment, I combined all 3 SIV targets that I've been talking about into a single reaction using 3 dyes rather than performing 2 parallel reactions with a common target. And then in a separate reaction, we test our host gene, RPP30. We have the 2 targets in RPP30 using the FAM and VIC dye. As you can probably see right away, those targets are much more abundant because of the host. So I think Holly and Lenny will appreciate me mentioning that switching from droplet digital PCR to PCR was really straightforward and required little optimization, especially thanks to some helpful suggestions from Holly that I think really allowed me to streamline the process, but it was a real easy change. So now that I knew the assay worked really well in the absolute Q, we of course wanted to do a head-to-head comparison of our 2 platforms to make sure we get the same results for a given sample, regardless of which platform we use to perform the assay. So we identified a small number of samples that we could use for this experiment. The first thing we noticed was the consistency of the partitioning with the absolute Q as shown in this graph here. So using the absolute Q, we averaged 20,440 partitions filled out of the possible 20,480. In the parallel experiment using droplet digital PCR, we averaged close to 12,000 droplets, which I would say is pretty normal for our experience. And I think it's worth noting too that not only do we get significantly more partitions filled with the absolute Q, but this is very reproducible, while droplet generation is much more variable. So in fact, you can see that I put the standard deviation for the droplets on this graph, as I did for the partitions for the absolute Q, but the error bars are so small you can't even see them on the graph. I also want to mention that in these parallel experiments, we had 5 out of 32 of our reactions fail with the droplet digital PCR platform. I'm considering failing anything below 7,000 droplets generated because we just can't trust that data. And actually, I think 7,000 is a pretty low threshold, to be honest. We did not have any reactions fail with the absolute Q. So overall, because we're able to interrogate more partitions with the absolute Q, and we're getting more data with the same amount of sample, which will hopefully, one, save us money, but also increase our sensitivity. So beyond being easier to use and more consistent, the absolute Q offers another advantage over the droplet digital PCR system that we have access to, which is that it can detect 4 dyes. The droplet digital PCR system that we have easy access to only detects 2. I know there are those that. So this gave us the opportunity to further multiplex our assay, as I alluded to earlier. With additional channels, we have the opportunity to combine all 3 of our SIV targets into a single reaction rather than running it as 2 separate reactions with that common target. So this allows us to be more efficient both in terms of our cost and in our use of sample. And again, it was really easy to make this change with little optimization required. I think I did 2 experiments because I varied the concentration of probe for one of the reactions. That really helped increase separation, but beyond that, it was very straightforward. So I'm actually going to show the results from my direct comparison of our 2 versus 3 targets in 1 reaction versus 2 reactions with that common target. So the column labeled original assay has our 2 SIV reactions, and the SIV multiplex has all 3 SIV targets in a single reaction. So importantly, the numbers in parentheses are the percentage of total partitions filled that were positive for each of the targets, which are listed individually. And I show this because this allows us to account for the different numbers of total partitions that were counted in each reaction. And as you can see, the results are almost identical for each target, regardless of how the assay was performed, which means that we can feel good about moving forward with our further multiplex reaction. So now I want to show you probably the most important results from our comparisons of the different variations of how we're performing the assay, and that is the amount of intact provirus per million cells that we found in each of these samples. So I'm showing results comparing the IPDA performed using droplet digital PCR in green, performed on the absolute Q with the 2 SIV reactions in orange, and in the golden color, we performed on the absolute Q, but put all of our SIV targets into a single reaction. In this experiment, we tested samples PBMC samples from 3 different macaques, one of which was SIV naive as a negative control here, and the other 2 were SIV positive. As you can see, the results from the 2 SIV positive samples compare very nicely across all 3 variations of the assay. Looking at the amount of intact proviral DNA on the y-axis, we can see that we get very similar results regardless of how we tested the sample. We did paired t-tests to confirm what we saw visually, that when we compare each of these different variations, there is no statistically significant difference from one another with a 95% confidence. So now that we know the assay works really well in the absolute Q, we wanted to have an idea of how sensitive the assay really is. So to do this, we performed a series of experiments where I spiked a known amount of an SIV plasmid into naive PBMC and tried to detect it. As you can see here, we can reliably and pretty accurately test down to 6.6 copies of the plasmid, which, as this assay was performed based on starting with 2 million PBMC, actually calculates back to 632 intact provirus per million cells down here. This was actually really encouraging, and I think it indicates that we're going to be able to detect even much rarer events because we did not even come close to reaching the lower limit of detection in this experiment. We also intend to increase our sensitivity by testing our sample across additional replicates on a plate, so that will be more sensitive, as Holly was talking about with pooling the data, which we will be doing anyway. So in a follow-up experiment, I just wanted to determine how few copies of that SIV plasmid we could detect per reaction. I'm used to doing a lot of qPCR, so I was curious how this would compare. So as you can see, as we get below 6 copies per reaction, we didn't detect it every time, but still more often than not, which I think is really impressive. I know qPCR, we wouldn't be seeing that, so we were quite pleased. So I'm just going to finish by showing you a little bit of the data that we generate using the IPDA. So because I'm part of the virology services unit, my group mainly does fee-for-service work, which means we actually have limited samples available for answering hypothesis-based questions of our own using this assay. But I just want to give you an idea of what the data looks like. So I will just show you some representative samples here. So this is what I imagine a sample report will look like that we share with clients. As you can see, we've calculated our DSI, the amount of cells that we tested in this reaction, and then most importantly, of course, the intact provirus per million cells. And you can see here too that we can indeed detect differences even though these animals were not distinctly different in any major ways. We have like treated and untreated animals available right now. Okay. So in conclusion, I just want to remind you that quantifying the latent reservoir of AIDS viruses is both technically challenging and critical for evaluating emerging therapies and interventions. The reservoir assays developed for this purpose need to be exquisitely sensitive. I hope I've convinced you that digital PCR, like droplet digital PCR, is uniquely suited for this particular assay and an ideal platform for performing the IPDA. This is again very evident when we think about looking for targets that are linked like we do with our SIV-OMV and POL targets. I hope that I've demonstrated that using the absolute Q instrument, we were able to enhance an existing assay by generating more consistent results and taking advantage of the opportunity to detect 3 targets in a single reaction. This has allowed us to both save time and sample using the absolute Q. And though I didn't show this visually, I can assure you that it is a lot easier to use the absolute Q than it is to generate droplets for droplet digital PCR. So with that, I just want to take a minute to thank everyone in my group, the Virology Services Unit and the Friedrich Lab. One thing that I didn't mention is that the analysis for the IPDA is actually fairly complicated and cumbersome. So Grace from my group has actually taken the time to write an R script to really streamline that process, which I very much appreciate. And Alex from my group does all my statistical work, so he's helped me out with that. I especially want to thank Dr. Matt Reynolds and his lab, who in particular, his grad student, Leah Mache, who did a lot of work in getting the IPDA up and running for our group, and also figured out the complicated analysis. Lenny and Holly really got me started with the absolute Q and made it real easy to switch over to using that. And of course, I am grateful to Molly and the Bio Garage for making the instrument available to us. And with that, I can take any questions. Very nice, Leah. So the Sci-5 for the 2 LTR um, probe was originally there wasn't as good separation, so I doubled that, and then also the RPP30, the VIC. Um, no, those were totally fine. I think it's just that Sci-5 is not as bright as the other two dyes, and for some reason it's the separation wasn't as good with VIC as it was with FAM in the host gene reaction. So I was just curious if increasing it a little bit helped, and it really did help to get rid of any rain that was in between the positives and negatives. Yes, sorry, I think I did label that one because no, you're absolutely right. It is. Mhm. It could be. That was on Droplet Digital, right? Yeah. Mhm. The same ratios or